Hello, I'm talking to you. This is Goddess of Science Biology, and I'm Mrs. Bolden. Today, we're finally getting to it. We're going to talk about the light-dependent reactions of photosynthesis in part 5 of my cellular energy series. Okay, so, alright, you can already see the bonus, so I guess I'm not going to be, surprising you when we get to the end there. But just ignore that for the moment. Alright, so remember we said that there are 3 parts to photosynthesis. Parts 1 and 2 are the dependent reactions, deep light dependent reactions. So in part 1, a pigment captures energy of a certain wavelength. So you probably remember from elementary science, Roy G. Biv, red, orange, yellow, green, blue, indigo, which is not really a color, and violet. Excuse me. So each one of those has a different wavelength of the waves that make up that light. So the pigments only capture energy that's of a certain wavelength and usually corresponds somewhat to color. So chlorophyll A, which is the primary pigment, captures blue and violet as well as red light. It does not capture green light. That's why plants look green, because the green is being reflected back into our eyeballs when we look at it. Well, if you have a chlorophyll A, you must also have a chlorophyll B, and that captures part of the green wavelengths. Not all of it, but some of it. So that's a secondary pigment. We also have other pigments called carotenoids, and they absorb the blue and the green and usually reflect back the yellow and the orange kinds of wavelengths. We know in the fall, particularly here in Maine, when the weather starts to change and it gets a little cool out, and just before the leaves fall off the trees, particularly the maples, all of the chlorophyll starts to break down, and then you can actually see these carotenoids in the leaves on the trees. That's why the trees are orange and yellow. You can't, the carotenoids are there, but you usually can't see them because there's just so much chlorophyll A and chlorophyll B. It kind of, the carotenoids get lost in all of that greenness. So the reason that it's kind of a good thing that you have 2 different kinds of chlorophyll and these carotenoids is because it absorbs more light over a broader range of wavelengths. It's not just one wavelength, it's actually a whole range of wavelengths. So it makes the plant much, much more efficient. Okay, so the sunlight energy passes from molecule to molecule until it finally gets to chlorophyll, and then chlorophyll goes, whoa! And the chlorophyll is actually attached to a membrane-bound protein. So that gets an electron excited from the chlorophyll, and it gets transferred to the protein that's kind of the anchor for the chlorophyll molecules. That energy gets passed on to other proteins, which make energy, which use the energy that has been captured and passed along to make ATP, our old friend, and our new friend, NADPH. So that's kind of part 1 in the nutshell. Part 2 gets a little more hairy. So we already have a little bit of ATP and some NADPH, but though we need more of that for part 3, which is coming up. So part 2 continues to use the energy to make ATP and to reduce NADP plus to NADPH. So we're going to make some more NADPH. So in the thylakoid membranes, there are these things called photosystems. You're like, what the heck is a photosystem? Well, I'm going to tell you. Photosystems are made of an antenna complex. You can kind of think of it like an umbrella. So the part of the umbrella that goes over your head in this situation is a network of chlorophyll molecules. And then you know, an umbrella is not really any good unless you have a stem or a handle to hang onto it. That's the accessory pigments there. So the energy passes from molecule to molecule until it gets to the reaction center. That would be the center of the umbrella. And then the reaction center is a transmembrane pigment complex. Okay, so it's kind of a weird thing. It goes through the membrane, that's where it's transmembrane, goes across the membrane, and part of it is protein, part of it is pigment. So if you think about this photosystem as the antenna is all the chlorophyll and other pigment molecules, and then the reaction center is the stem or the handle of the umbrella. What happens when it gets to the reaction center, it transfers the excited electron, not really the energy, to an electron acceptor molecule. In chloroplasts, it has two photosystems, and if this is not the craziest thing you've ever heard, I don't know what it is. So we have photosystem 2, and you can abbreviate it with PS2. That was actually discovered second, but when the electron gets transferred to the photosystem, it doesn't go to photosystem 1. That's down the system a little bit. It goes to photosystem 2. And the reason that this one wasn't photosystem 1 is because it was discovered second. So it's just weird. Scientists are just kind of odd. I don't think that they actually called the first one photosystem 1 because that would mean that they knew about 2. But once they had to number them. Okay. So photosystem 2, what that does is it breaks apart a water molecule to supply some electrons to photosystem 1. So that's kind of the purpose of photosystem 2. It's just like a supply truck for photosystem 1. And speaking of photosystem 1, it gets the electrons from photosystem 2. Great. And then it transfers those electrons to our friend NADP plus to make NADPH. Awesome, because that's an energy molecule as well. It's kind of the ATP's smaller, weaker cousin. So the electrons from photosystem 2 passes to something called the B6F complex, which is kind of weird. And that just pumps the hydrogen into the thylakoid space, and that becomes a little more important later. Okay, so what's going on in photosystem 1? It gets the electron from photosystem 2 and transfers it to make some NADPH. Great. Also, the electrons from photosystem 2 once it gets to the end there, it also gets passed to something called the B6F complex, which pumps hydrogen into the thylakoid space. Okay, so once the hydrogen, you're loading up on hydrogen in the thylakoid space, obviously it's going to make a higher concentration of the hydrogen. And this is where we have a new term here. It's called chemiosmosis, which is, there's a molecule called ATP synthase, okay? Synthesis means to make, so what's going on here is ATP synthase it's an enzyme, uses the hydrogen differences between the sides of the membrane to make some ATP. So ATP synthase is an enzyme that makes ATP. They kind of named it in a sort of logical fashion. So this is why it's really important that the hydrogen ions get pumped into the thylakoid space. So the ATP and NADPH from the light-dependent reactions go on to the Calvin cycle, which is sometimes called the citric acid cycle. It's a little bit weird. So this is the Calvin cycle. So some stuff going on here. And we'll talk more about that later. Thanks for joining me. Have a great day.